All right. Good afternoon, everyone. It's so exciting to be here for this very, very interesting session today, matters pertaining to unproven cellular therapies in South Africa by Prof. Michael Pepper. We are waiting in anticipation to learn a lot from the expert in the field. I see there are already 55 people online. So because I really want to give Michael the floor, for us to learn. Let us just continue. Okay. So this is the deep dive management team. It has changed a bit in the last couple of months. We know Prof. Estelle Verberg, who's the team leader, and we have Terrence Solomon, Michaela Brooks, and Melinda King, which have joined in the last few months, dealing with a combination of administration and medical related matters. So thank you very much to this team for setting this up and for running such a professional show. Okay, just so that you know, everything that we do in Deep Dive is recorded and it can be found on the Blood SA website under resources and then Deep Dive. Please, can you all mute yourselves during the presentation? And as Michael is speaking, please add your questions in the chat box and we will respond to them at the end. But usually there's a large number that attend, so please ensure that your microphone is off. All right, so you can get CPD points for these sessions, but you have to attend for a minimum period of 1 hour. Please make sure that your HPCSA number is entered correctly so that the points can be allocated. And if you have any questions in this regard, please email admin@bloodsa.org.za. Okay, you can also join the mailing list to sign up to receive invites for the deep dive sessions by scanning the QR code that is on your screen at the moment, or you can visit the Blood SA website, go to resources, subscribe to Deep Dive. And at the end of the session, you will be getting a feedback questionnaire. And we really ask and encourage as many people as possible to fill this in and to be as honest as possible. Okay, if we look at upcoming events, very excitingly, in just 2 weeks' time, less than 2 weeks actually, we'll be having the SASET SASBO conference at the Sandton ICC. It is still possible to register. Maybe they'll fit you in, although it's closing in the next couple of days. So we have an extremely exciting agenda on all 3 days. So we want to encourage as many people as possible to come. Also allows us to just catch up with each other. And as this only happens once every 2 years, we also want to invite you to register for the Cell and Gene Therapy Conference hosted by the ICMM, Institute for Cellular and Molecular Medicine at the University of Pretoria, of which Michael is the director. This is taking place from the 27th to the 29th of October this year at Future Africa. And you will receive the invitation in the chat box. The abstracts, there are many categories within the cell and gene therapy field, and we really want to encourage as many people as possible to not only submit abstracts, but also to attend. We also are very excited to say that we now have a space on the Blood SA website where the manuscripts or white papers that we are writing within the Cell and Gene Therapy Working Party are accessible. The first is the publication from last year, Progress in Advanced Cellular and Gene Therapies in South Africa and Barriers to Patient Access, which we wrote as a consortium. And since the regulatory matters are probably some of our biggest concerns in this space in South Africa, we also have a white paper for sustainable regulatory framework, for advanced therapeutic products in South Africa, also available in the public domain for you to read and learn from. Okay, next week in Deep Dive, we'll be talking on the clinical relevance of HLA-DPB1 T-cell epitope matching in donor selection with case studies, something very relevant to our clinical practice. So I hope it will be a jam-packed session again. Karen, I don't seem to be able to go on to the next slide. Okay, thank you. Okay, so we will shortly start with the session. As I said, please put your questions in the chat box. At the end of the presentation, we're going to have a nice discussion about this very interesting topic, and we will close after that. Okay, so I'm not sure if there is anybody in South Africa in this field that doesn't know Prof. Michael Pepper. And he is so distinguished that I want to start with the fun things first, that he is in fact an international ballroom, how do you say, champion? He is winning international ballroom competitions, and he's just been appointed to the Gauteng show jumping team. So certainly Michael is teaching us how to do life right by being an exceptional scientist, while maintaining his hobbies at an extremely high level. I also know Prof. Michael Pepper personally. He was my supervisor of my PhD, and can certainly say he is exemplary, not only as a scientist, but also as a human being. He has made substantial contributions in cellular and molecular medicine in the fields of vascular biology and immunology. He's held permanent positions at both the universities of Pretoria and Geneva in Switzerland, where he spent 20 years. He's currently the director of the Institute for Cellular and Molecular Medicine and the SAMRC Extramural Unit for Stem Cell Research and Therapy at UP. He has more than 400 medical and scientific publications and has an H index of 79 and 98 in Scopus and Google Scholar, respectively. He's a member of the Academy of Science of South Africa and has received a number of awards for his research, including the South African Health Excellence Award for Scientific Excellence in 2019. In June 2026, he was ranked number 2 in South Africa and 1,711 in the world in biology and biochemistry by research.com. His work has had an impact on the development of cell and gene therapies in South Africa, and he continues to be a leading figure in addressing ethical, legal, and social implications of research in these areas. So on that incredible note, I think we can certainly say we are learning from the best today. So over to Michael to teach us really about his expertise in the field of unproven cellular therapies. Thank you, Michael.
So thank you very much, Candice. That was not all necessary, but highly appreciated. Thank you. I hope my screen is visible to everybody.
Yes, all good my side.
Okay, good. Thank you. So thank you. This is really a wonderful opportunity because it gives me an opportunity to talk about things that are of great importance to the global community, but also in South Africa. And I'd like to begin by just sharing these declarations. So I have received funding for the work that we do from all the funders that are listed there. I've also been involved in the creation of 2 startup companies, Antion and Altera Biosciences, of which I'm a co-founder and director. And I do have a, if my slide would just advance, I do have a disclaimer I'd like to make before I start. So I'm a non-practicing clinician involved primarily in biomedical research based at the Faculty of Health Sciences at the University of Pretoria, as Candice has mentioned. My titles are listed there. I've listed my degrees at the bottom of the slide, and the reason for doing all of this is simply to state that I make no claim to being a legal expert. I am going to talk a little bit about legal matters in South Africa, but I would like you to consider these as being my personal views in the context of today's discussion. So we have the great privilege as medical practitioners of being able to treat our patients for a variety of diseases. And at the start of our careers, we bind ourselves to a number of oaths and commitments. The dominant one globally, that is the successor to the classical Hippocratic Oath, is the World Medical Association Declaration of Geneva from 2017, called the Physician's Pledge, which essentially focuses on the idea that the health and well-being of our patients will be our first consideration. In South Africa, the Hippocratic Oath was replaced long ago by university declaration ceremonies every year from graduating students. We're bound by HPCSA ethical and professional rules, and then beyond that, also by constitutional and human rights frameworks. So this then provides the foundational pillars of clinical practice, whether it be in South Africa or elsewhere. Care is patient-centered and decision-making is shared. So that respects patient autonomy. We abide by professional ethics. We take great cognizance of scientific evidence in the form of evidence-based medicine that addresses the principles of beneficence and non-maleficence. And we also agree to adhere to legal and regulatory imperatives. So those three, autonomy, beneficence, and non-maleficence, are three of the four pillars that underlie medical ethics. The fourth being justice, which obviously is interlaced with all of the above. So today's talk is about unapproved therapies, and the overarching framework that we work under when it comes to advanced therapies is shown here. So we distinguish advanced cellular therapies from gene therapy, and these advanced cellular therapies include regenerative medicine and adoptive therapies, whereas gene therapy treats or prevents disease by correcting the underlying genetic problem by altering a person's genetic makeup instead of using drugs or surgery. So the main focus of my talk today is going to be on regenerative medicine. I will touch on the other two as we go along. For completeness sake, under adoptive therapies, we have things that we're familiar with, certainly in this audience, namely CAR T cells, and the other adoptive therapies are listed there. And then in gene therapy, we consider gene replacement, gene addition, and gene editing. So let's turn our attention now to regenerative medicine. This is a fairly exhaustive classification of what we would consider to be components of regenerative medicine. So there are four components: stem cells, orthobiologics, scaffolds, and then prolotherapy. Stem cells are either adult or pluripotent in nature, and the ones I think that most people are familiar with in blood assays, hematopoietic stem cells, but we also have mesenchymal stem cells. We have two classes of pluripotent stem cells, namely embryonic and induced pluripotent. Orthobiologics include platelet derivatives such as platelet-rich plasma, platelet lysate, and growth factors. Scaffolds are essentially matrices. And prolotherapy involves the injection of an irritant into tissues locally in order to induce an inflammatory and hopefully a beneficial healing response. Now, the one I think most people on this call are familiar with is hematopoietic stem cell transplantation, which is universally approved. The indications vary broadly: cancer, blood disorders, and genetic disorders. And what you can see on this slide is a hematopoietic stem cell on the left, undergoing differentiation to give rise to all of the white blood cells, red blood cells, and platelets that are needed in our bodies. Is this the only form of universally accepted regenerative medicine? The answer is no. So there's been great progress in this field. As I've said, stem cell or bone marrow transplantation is standard of care worldwide. The evidence is excellent. Then cultured epidermal autografts, which is used in certain burn centers, the evidence is good. Limbal cell transplantation in the eye, again, in specialist ophthalmology care, the evidence is good. Bioengineered skin substitutes for selected indications, again, the evidence is good. And then there are some where there's less well accepted evidence, but nonetheless, these are there. This is ACI, autologous chondrocyte implantation for cartilage defects and other cartilage products, and some selected corneal products. So I think these are things that have been through multiple rounds of clinical trials in different parts of the world and are universally accepted as being so-called regenerative medicine therapies. There are, however, a lot of other so-called regenerative medicine therapies which are not universally accepted for routine clinical use. I've listed these here. So stromal vascular fraction. This is prepared from adult tissues, most commonly from adipose tissue. Mesenchymal stromal or MSC therapies, adipose-derived stem cells, bone marrow aspirate concentrate, PRP, exosomes and secretome products, prolotherapy, and then tissues that are made available around the time of birth, so amniotic or Wharton's jelly products. So I list these because they are used in some settings, but they're not universally accepted in terms of routine clinical use. So today's topic is about unproven therapies, and why does this matter? So let's first define unproven therapies. So these are products that contain cells, tissues, or their derivatives, marketed or administered without sufficient evidence of safety and efficacy for the intended indication and or outside applicable regulatory requirements. So this is a very clear and clean definition. And to complement this, many products are marketed as so-called stem cell therapies, but contain few or no viable stem cells, which does not really qualify them as stem cell therapies. The problem is related to the fact that there is a very rapid growth of commercial clinics globally that offer these therapies to patients, often at considerable cost. And this is considered by many to be an exploitation of patients that are vulnerable. So if you have a chronic incurable disease, you are vulnerable. And when the so-called white coat effect, with this power differential between someone of authority wearing a white coat tells you that something is going to work, you believe them. And there's also financial exploitation because most of these therapies are not covered by medical aid and are offered in the private sector where people have to pay out of pocket. The other reason this matters is that there is well-documented harm from these unproven cellular or so-called stem cell therapies. So let's go through that. We can define 3 layers of harm. So the first is direct biological or procedural harm. And what I've listed here is very well documented in the literature from multiple sources. So let's go through these. The first is severe infections, from local abscesses to sepsis and septicemia, The most common cause for this is when manufactured products do not comply to GMP, that is good manufacturing procedure protocols. Then thromboembolic events like pulmonary embolism, myocardial infarction, cerebral infarction, stroke-like syndromes. These are most commonly seen when cellular products such as SVF, stromal vascular fraction, are given intravenously. Then ocular injuries. So you may have up to bilateral permanent blindness, and this usually occurs after intraocular injection of these therapies. Neoplastic cancerous growths and ectopic masses. This occurs when pluripotent cells are injected, and these cells are typified by their ability to proliferate in an uncontrolled way. Then there's neurological injury, so paraplegia after spinal injections, glioproliferative lesions after intrathecal or IV administration, and multi-organ failure and death have also been reported, particularly after systemic or intrathecal administration. So these are layers of harm that certainly are very concerning from a biological and procedural perspective. But there are other layers that one needs to think about. The second one is systems level harm. So many unproven interventions are delivered outside of formal trial governance without rigorous manufacturing controls, sterility validation, dose justification, biodistribution data, adverse event capture, or long-term follow-up. So these are the things that would normally characterize the introduction of a new medicine into the market, usually through the process of conducting a clinical trial. Then there is also a third layer, which I refer to as epistemic and economic harm. So patients are exposed to misleading direct-to-consumer advertising, therapeutic misconception, often when signing an informed consent document, and as I mentioned, substantial out-of-pocket costs. And I'll give you an example of this a little bit later. And the problem is that in addition to affecting patients, this can delay evidence-based care and distort public trust in what are legitimate regenerative medicine options. So this then creates a negative perception of the field, which is not helpful when trying to introduce beneficial new therapies into the market. So in order to combat some of these problems, a global regulatory framework has evolved over time, over a long time actually, several decades, and from major jurisdictions around the world is applied to all cell and tissue-derived products. So there are three elements. The first concerns origin. In other words, is the product autologous, that is from the patient's own tissue, allogeneic, that is from a donor, or xenogeneic, that is from another species. Secondly, manipulation. Is the product minimally manipulated, or does processing exceed that threshold? And then third is the site of use. Is the product applied to the same anatomical function? In other words, it's homologous, or is its application non-homologous? That is to say, it's administered to an anatomical site which is not commensurate with its normal function. So I'm going to go through each one of these in some detail now to explain to you how this global regulatory framework has evolved. So let's talk first about harm that is related to origin. So when it comes to allogeneic or xenogeneic products, obviously the risk profile changes considerably compared to autologous products, because these products are recognized by the recipient's immune system as being foreign, they're incompatible, they are rejected, and if introduced into a small space, may even cause local inflammation as a result of cell death, which may be detrimental to the patient. And of course, with xenogenic products, we have the risk of the transmission of infectious agents from other species, not normally found in our own species. So there is a need when applying allogeneic and rarely xenogenic products for close monitoring and to ensure that donor recipient matching is carefully monitored. And so source is not a trivial detail, it is a core safety variable. Let's go now to harm related to manipulation. So in all major jurisdictions, the notion of minimal manipulation can be defined as processing that does not alter the original biological characteristics, structural properties, or the intended function of cells or tissues. So what would that be? Examples would be washing, centrifugation, cutting, sizing, and freezing. So you're not actually altering anything that is biological related to the product. What constitutes more than minimal manipulation is processes that alter these biological characteristics, structural properties, or intended functions, which result in a substantially modified product. Examples would be enzymatic dissociation of tissue, expansion of cells in tissue culture, activation of cells, differentiation, and of course, genetic modification. The first one, enzymatic dissociation, is sometimes considered as being trivial, but let me just remind you of the basic principles of cell biology, that a cell is linked to the extracellular matrix via transmembrane proteins called integrins. And when this association is disrupted, the pattern of gene expression in the cells changes very rapidly. And so these cells lose their basic biological characteristics and become something else. So globally, therefore, manipulation has become a major threshold for oversight. In other words, everything that is more than minimally manipulated should be then subjected to oversight from a regulatory perspective. Okay, let me come to the third one then, harm that is related to site of use. So homologous use would be use of cells or tissues to perform the same basic function in the recipient site as in the donor site. In other words, it performs its natural function after transplantation. And examples would be skin used to replace skin, or bone used to replace bone, or fat transplanted to a natural site where it would function as fat would function normally. And what constitutes non-homologous use? So this is where cells or tissues are used to perform a different basic function at the recipient site than at the donor site. So its biological function is different from its natural role. And what are a few examples? An example there would be adipose tissue or its derivatives that are processed and then used to treat osteoarthritis, such as, for example, being injected into the knee, and also used to treat neurological disease. So therefore, non-homologous use is also considered to be a major threshold for requiring regulatory oversight. So I've been over these three components now, and not adhering to these components amplifies the risks of harm that we have discussed already. So that pertains to non-autologous material that is more than minimally manipulated, and material that is used in a non-homologous fashion. In most cases, there is no dose finding. There is no adherence to either determining toxicology or biodistribution. In many cases, GMP level manufacturing or sterility control are not instituted, which renders these products high risk in terms of transmitting infectious agents. And some of the routes that are used for the administration of these products are also risky, such as intrathecal, intraocular, intravenous, as I've mentioned previously. And most importantly, in many of these cases, there's no systematic adverse event monitoring or reporting. It is very rare to see from so-called stem cell clinics operating around the world any systematic reporting of adverse events from their patients. So I'd like to give you an example of work that we did a few years ago, published in 2019 in the South African Medical Journal. We went around the country and identified patients who had spinal cord injury. So the data is all shown here. We had 8 males, 3 females, 4 had paraplegia, 7 were quadriplegic. All had received so-called stem cells for their spinal cord injury. And the nature of the cells they had received here, in 3 cases were autologous. In 1 case was allogeneic cord blood. Many were xenogenic, so 4 were from rabbits, 2 were from sheep, 1 was unknown. The clinics at which these treatments were administered were in Germany, the Netherlands, India, China, and in South Africa. The route of administration of these therapies were oral, subdermal, intramuscular, and intrathecal. Now, we did very careful analysis and assessment of these patients, and there was no improvement in any of these patients. And to the patient that received intrathecal treatment, the symptoms were actually worse after the treatment than before. The patient had gone up a grade in terms of severity. And when examining the cost of these treatments, the average cost per treatment was over 150,000 Rand. Travel, as you can see there, and the total then for those patients that did travel was at around 200,000 Rand with no benefit. So I think this is a very good example, and there's certainly been a major motivator for us to look into the problem of unproven so-called stem cell therapies. Okay, so I'm going to come now to the legislation in South Africa, and I wish to repeat my disclaimer. I'm not a legal expert, so my interpretation here is personal, based on my understanding of the legislation in the context of today's discussion. So the first important piece of legislation is the so-called Medicines Act, Act 101 of 1965, which defines a medicine broadly as a substance or a mixture of substances used or intended for use in diagnosis, treatment, mitigation, modification, or prevention of disease, in restoring, correcting, or modifying bodily function. And the uses are shown there. So diagnosis, treatment, mitigation, modification, restoring or correcting any somatic or psychic function. So that's the broad definition of medicines, and it does not include specifically the products that we are talking about today. However, the South African Health Products Regulatory Authority does have a category that is defined in this act as a biological medicine. SAPRA has a website. in which they define biological medicines as products produced from living organisms that contain components of living organisms. So this includes a variety of biological medicines derived from humans, animals, or microorganisms using biotechnology. And the SAPRA guidelines list some of these here. And they do specifically state that they include, but are not limited to, the following. So plasma-derived and animal products, vaccines, biotechnology-derived medicines, and products developed for human gene therapy. So although the advanced therapies that we are speaking about today are not explicitly mentioned in this definition, they are nonetheless biological medicines because they are produced from living organisms and they contain components of living organisms. So all the therapies that we're talking about today are indeed covered by this definition of biological medicines, which comes from Act 101, as I showed previously, and are listed on the SAPRA website. SAPRA has in fact established a biological medicines unit, and this unit is responsible for the evaluation of applications for the registration of biological medicines, for amendments to these biological medicines, for vaccine lot releases. Other activities include communication with the pharmaceutical industry on policy matters, and the establishment of a framework for the regulation of blood and blood products, and stem cells and cell-based therapy. So SAPRA does have a unit, and many people on this call who are involved in some of the exciting new work involving adoptive therapies have had to go through processes of registering their products and their clinical trials with SAPRA. So I think the notion that advanced therapies exist in a regulatory vacuum in South Africa is not true. This is not a correct concept. And in fact, these medicines have been regulated for a long time. The Medicines Act has been around since 1965 and does contain these different categories considered to be biological medicines, which have been included in SAPRA's guidelines. As you can see here, version 3 was from August 2014. So they have been there for more than 10 years, governed under the SAPRA Biological Medicines Unit. So what are the implications? So first of all, with regard to registration, a biological medicine must be registered before being marketed. In other words, before being communicated to the public via a website or other forms of marketing. The holder of the registration is responsible for all aspects of the product, including safety, quality, and compliance with the conditions of registration. The guidance emphasizes that changes in process, packaging, labeling can affect the product's performance. So the registration is about the totality of the product's development and its subsequent use. So in practice, that means that the dossier must address the identity, manufacture, characterization, comparability, and risk management at a higher level of detail than is required for a simple chemically defined medicine. Most importantly, small changes in manufacture can result in large changes of function. Okay, what are the implications as far as clinical trials are concerned? Well, simply because a biological medicine comes from a human tissue, blood, or other living source does not mean that it is exempt from oversight. For products that are not yet registered, whose clinical utility is uncertain, use in patients should begin in a clinical trial and not in routine clinical practice. And in addition, the regulatory framework requires that the studies intended to ascertain safety, efficacy, or performance occur under approved pathways with both ethics and regulatory control. So when entering into a clinical trial, approval from an ethics committee and from a regulatory authority is required. And with regard to the other implications, biological medicines are not only regulated at the point of first approval, they remain under oversight throughout their life cycle. In other words, it extends beyond registration and clinical trials. So ongoing evaluation of variations in the product and post-registration changes. So this would include manufacture, quality assurance, adverse event reporting, control over import and export, ongoing pharmacovigilance, and advertising restrictions. Okay, I'd like to turn now to GMP manufacturing. And I know also that some of the people on the call have recently gone through the process of obtaining GMP authorization for manufacturing products in South Africa. Not a trivial event, but absolutely necessary. So this is the manufacturing benchmark that aligns internationally with best practice. And in fact, the SAPRA guidance is aligned to PICS, which is the international framework that harmonizes GMP across different jurisdictions. So why GMP? Because without GMP, the identity, purity, potency, sterility, and traceability of the product cannot be reliably ensured. So let's go through the three elements that I mentioned earlier as far as manipulation is concerned. Once a product is processed beyond its basic collection and use, in other words, it's more than minimally manipulated, the risks of contamination, instability, and loss of potency are real and required regulatory oversight and controlled manufacture. Again, products from different sources, autologous, allogeneic, and xenogenic, have different risk profiles. This needs to be controlled for. And particularly with non-homologous use, so when taking cells from one anatomical localization and using them in another, this requires much greater assurance that the product is safe, consistent, and fit for the claimed purpose. I think one of the things that we don't give enough importance to, we focus on safety, but it is really important that if you're going to propose a therapy, there must be a reasonable chance of this therapy working. So efficacy is also very important. So GMP manufacture, certainly in the case of many therapies that are profiled in the public domain and would qualify as unproven therapies, are not subjected to GMP manufacturing. And I guess it would be up to the legal authorities, the court, to determine whether in fact this is an infringement on the local requirements of South African law. Practitioners and health facilities also need to be registered. So health practitioners must be registered with the Health Professionals Council. And as far as regenerative medicine is concerned, in a discussion with Candace recently, I learned that in order to perform hematopoietic stem cell transplantation, you need to either be a medical oncologist or a clinical hematologist. But for other forms of regenerative medicine, such as, for example, the use of stromal vascular fraction, there is no specific requirement at present. And so this begs the question, should there be some form of requirement that is legislated in order to be able to practice these forms of therapy? Facilities also need to be regulated. This is covered in the National Health Act. So health establishments need to be registered. And the National Health Act also regulates the removal of and use, processing, and control of tissue, blood products, and gametes. So again, there is legislation in South Africa that requires all of these requirements to be met before one can embark on many of these therapies. And then just as far as the South African Constitution is concerned, Section 233, I will quote this verbatim, says that when interpreting any legislation, every court must prefer a reasonable interpretation of the legislation that is consistent with international law over any alternative interpretation that is inconsistent with international law. I think that speaks for itself. And Section 39, which pertains specifically to the Bill of Rights, requires courts, when interpreting the Bill of Rights, to consider international law and may consider foreign law. So what is the implication? The implication is that where South African statutes do not expressly codify the three things we mentioned, source, manipulation, and homologous use, these constitutional provisions nonetheless support reliance on internationally recognized best practice, both in terms of safety and in terms of human rights. There's an additional layer when it comes to international guidance. The International Society for Stem Cell Research and the International Society for Cell and Gene Therapy have very detailed regulations concerning the development and the application of cell and gene therapies. Although they do not have legal force in South Africa, they provide an important benchmark against which clinical practice, research conduct, and patient counseling may be evaluated. They underline that patient welfare and transparency in all communications are core principles, and they provide very detailed guidelines as far as processing, manufacture, and conduct of clinical trials is concerned, all of which need to be performed under expert independent review and oversight. A question I'm often asked is, must South Africa always conduct its own clinical trial? In other words, if a product has been shown to be useful elsewhere, does it have to go through a clinical trial in South Africa? In other words, under what circumstances is internationally generated evidence sufficiently robust and transferable to justify implementation in South Africa without the need for a clinical trial? So this is not an easy question to answer. Arguments for clinical trials are shown there. So there may be a different disease phenotype. Local genetics are certainly different. There may be different comorbidities, standards of care. Local manufacturing processes may be different, as may the processing methods of materials that are extracted from donors. Arguments against clinical trials include unnecessary delay and limiting patient access to these very important therapies, duplication, ethical concerns about repeating studies where uncertainty is no longer present, obviously cost, and opportunity cost. So we're missing out on an opportunity to provide our patients with things that could be hugely beneficial. So one way of looking at this, and I'm not going to go into this in detail, it's a separate discussion. There are several very important algorithms that have been used to look at the potential benefit of therapies that have been tested in many other jurisdictions. And the general comment I can make about the discussion today is that many of these studies are vague, underpowered, and do not have clear endpoint criteria that are comparable across different studies. But one does nonetheless need to bear this in mind, and there may be some therapies which we may be able to introduce into the market without a clinical trial. But if they meet the criteria that I mentioned earlier, they will nonetheless need to be registered with SAPRA. So another question that often comes up is the tension that exists between innovation and regulation. And it's often suggested that regulation is the enemy of innovation. In other words, regulation prevents innovation from occurring. And I would like to argue that this is not the case. The two are complementary. So regulation requires that the product and the manufacturing process and the claimed use be aligned with evidence and oversight. And the overall aims are simply to protect the patients and ensure efficacy, and also to protect the field from reputational damage. This ensures sustainability. So if one was to introduce a product and it was to cause harm because it had not been adequately overseen, this would definitely reduce the sustainability of that product. So here's the central thesis. South African healthcare professionals are bound by South African law. If not codified into South African law, or where the law lacks sufficient granularity, international law and best practice from all these entities as shown here, so FDA, EMA, World Health, ISCT, ISSCR should serve as guidance. Some practitioners and commercial providers, however, interpret the absence of codified directives as permitting the marketing and clinical use of interventions for which robust evidence of safety and efficacy is lacking. And I would like to suggest that regardless of the interpretation adopted, healthcare professionals remain bound by their professional and ethical obligations, which I referred to right at the beginning of my talk, which are to prioritize patient welfare, respect patient autonomy, obtain informed consent, and practice in accordance with the best available evidence. So let's look ahead. Let's look ahead with confidence. What does all of this mean? Our aim in all of this is to produce a comprehensive, evidence-based framework for the responsible clinical translation of advanced therapies. And I'm delighted to say that SAPRA is embarking on multiple parallel processes to ensure that this aim is achieved. So in summary, a biological medicine once brought within the regulatory ambit of the Medicines Act and SAPRA guidance is subject to layered oversight, pre-market registration, ethical and regulatory approval for a clinical trial, and then continuous controls thereafter over manufacturing, quality, labeling, adverse event monitoring, and any product variations. Why this level of security? For biological products, these factors, in particular manufacture, are integral to the identity, safety, and efficacy of the medicine and may change over time. And where the law is silent, science, ethics, and professional integrity remain. Legal permissibility per se, therefore, does not establish clinical validity if these other factors are ignored. So thank you for the opportunity, and I'm very happy to take some questions.
Wow, Michael, you did not disappoint. I have about 1,000 questions, so I don't see anything in the chat as yet. So I'll start us off while people are brewing about this newfound knowledge. I'm very interested to know how we actually surveil these clinics. So, of course, people who are administering these therapies are not on this call. It never happens like that. How do we actually find out, or even in your publication, was it only when these patients came forward with complications that we actually became aware of these clinics, or is there a better way for us to identify who is providing these unproven therapies?
Candice, thank you. I did actually have a slide which I wanted to put in, but then I just did not want to expose myself to unnecessary criticism. But it's very, very easy to go. You just go onto the internet and you'll find that there are probably half a dozen clinics in South Africa that are offering allogeneic, more than minimally manipulated therapies that are not used in a homologous sense. We did have one or two people in the stem cell paper for spinal cord injury that did approach us, and that's what initiated the study. I certainly have not been approached by people who've been treated by these clinics that have had adverse events. I do know many people who have been to these clinics, many of them who've written to me or contacted me in a professional capacity, and some in a personal capacity. I don't know whether they're registered with SAPRA, and I'm certainly not pointing a finger at them. However, I think it would be an easy exercise for SAPRA to identify these clinics and to approach them. And if they're willing to adopt the format that has been proposed, I think it'd be very easy to see whether or not these clinics should be subjected to regulatory oversight.
So from these clinics that you're aware of, if you are aware of some, where do they find their source material? I mean, who is providing them with the stem cells? Is it being imported? I mean, if you're saying allogeneic, is it imported, or are we manufacturing it locally? Because if it is imported, how is it getting through customs? How is it getting into the country?
So both approaches are being employed. So there are stem cells that are being imported. And I'd like to say this is not just for humans, but across other species as well. So in the veterinary space, this is also being done. And so these are allogeneic. Very often they are non-homologous. So they come from one source and are being used in another source. That would almost certainly guarantee that they would be rejected by the recipient's immune system. And if this is in a confined space like a joint, the products that arise from the process of cell death would almost certainly create inflammation, which would aggravate rather than improve the problem that is being addressed. So in addition to these allogeneic therapies, I believe these clinics, based on what they put into the public domain, are extracting adipose tissue, extracting fat from patients, and then subjecting them to enzymatic digestion, sometimes to culture expansion of the cells. So they would definitely qualify as being more than minimally manipulated. And these cells are then being administered in a non-homologous fashion.
Okay, very scary to know that these things are actually happening. I think what you mentioned, and we have discussed this previously. Anybody else who wants to talk, please put up your hand, because as I said, I have 1,000 questions, so the floor is really open. You can put it in the chat or you can speak. Is that in the absence of formal guidelines, there are always gray areas, and these gray areas are taken advantage of. So we at present have genetic therapies being delivered to patients. CAR T cells here. Prof. Johnny Mahlangu, who I saw was online, but there are so many people, I don't know if he's still on. And we have the National Cell and Gene Therapy Working Party, and it's probably time for us to develop some kind of a framework within which or at least we make a statement to say, as the National Cell and Gene Therapy Working Party, we advocate for the following, that if you want to manufacture cellular therapy products, you have to have, or at least be developing a GMP facility. We at ACT do manufacture CAR T cells, and it's rigorous, the clean room facilities that are required. And as you've mentioned, none of these clinics are probably adhering to any of these rules, which is probably part of the problem why these patients deteriorate, etc., or at least a part of the problem. And besides that, probably provide some kind of a hospital exemption to say a patient receiving CAR T cell treatment must be nursed within a hospital that has transplantation facilities with ICU facilities on site. Now, we do not have anything like that in place as a group of clinicians on the clinical part. And on the manufacturing side, we certainly don't have guidelines either. But just listening to this talk, it is probably time that we try and guide these kinds of things. Whether or not they will be adopted as policy, that's another story, but at least put out a statement. So do you want to comment further on that? And I see there are some members from the Cell and Gene Therapy Working Party on the line regarding the manufacturing, and then some transplant clinicians as well, if you want to comment.
Candace, so in digging deep into this, as I've shown you, there is a lot of legislation in place in South Africa that covers many of these areas. And I think we simply have not been aware of the way in which this legislation must be applied. We've not been applying it, even though it's been there. So to complement that, what we would need is a set of guidelines in some of these areas. And I'm very excited to say that SAPRA is in the process of putting together various groups that will specifically address this going forward. I'm also aware of the fact that the Medicines Act is currently being revised. So we can expect for there to be changes which will directly impact what we're talking about. What's happened in the past with other areas that I've worked in is where there has been the absence of local legislation. As a group, we have created our own standards and subjected ourselves to self-regulation. And in every case, every member of the community has been willing to do that. What would be really exciting is if some of this material was then adopted by SAPRA and other bodies, once it's been proven to be accurate and implementable. So I think we're on our way, and I know in Blood SA we have discussed this extensively, but I think there is now, you're quite right, the time is now right for us to start putting into place things that can assist practitioners, and that will hopefully be endorsed by government at some later point.
Is there anybody else that wants to make a comment at this point? Candace, Murray. Candace is sitting behind me. Yeah?
I'm trying to find the raise hand icon.
I knew you would have something to say.
So just to say thanks to Prof. Weber for an excellent talk once again. And just, yes, that this, we are very encouraged by SAPRA's direction, and our experience during our recent GMP audit towards getting our facility licensed for the manufacture of GMP cell therapy products was extremely enlightening. As a JC accredited unit, we thought we had every regulation under control, but there are actually a number of things that a GMP cell therapy facility should take into consideration in its PICS guidelines, and the SAPRA documents are particularly helpful. But we experienced genuinely a push towards compliance and a helpful hand from SAPRA rather than sort of an exclusionary attitude from SAPRA. So it was very encouraging. So as Prof and Prof mentioned, it really is the time now to push towards compliant cell therapies and to expose these people who are unfortunately riding on the coattails of sort of miracle stories from licensed facilities and thinking that stem cells can come from absolutely anybody's backyard.
Okay, thank you for that, Candice. For those who don't know Candice, she is the manager of the processing facility at ACT and was pivotal in starting up the CAR-T trial here in our lab. I see Krishna's hand is up. Krishna, over to you.
Thanks, Prof, for an excellent talk. I don't know how to formulate this question, but what's a company that supplies devices to these people that provide the cell therapies, that prepare the cell therapy product? You know, they like the closed systems. Do they have any liability in order to do so, to make sure that they provide these devices to people that can use them responsibility and have the authority to use them?
So thank you, Krishna. That's a really important question. The answer is yes. So the Consumer Protection Act is very clear on the process of strict liability. And strict liability means anybody in the manufacturing chain can be held liable for something that may go wrong with a patient. So take, for example, somebody who has a hip replacement. Anybody along the manufacturing chain, including the people who manufacture the hip that is being inserted, should there be a problem at a later stage, they too could be liable. So the answer to your question is yes. Our Consumer Protection Act covers that. It's not just the practitioner that's administering the therapy. It could be anybody along this chain of supply that could be incriminated should something happen.
Thank you.
Anything else, Krishna?
No, that's all. Thanks. Yeah.
Okay, so I would like to know if we are approached by a patient that says, this clinic is offering me, stem cells for my spinal cord injury or my child's cerebral palsy, etc. What are the channels through which we should be reporting this? Or we know we have a responsibility to report it, but who do we report it to?
So that is a problem. Unfortunately, there's no official body. I would suggest that the most appropriate way of approaching this would be to report it to SAPRA. I don't want to badmouth SAPRA. I think they're doing a fantastic job. They took over from the MCC, and they've been operating under difficult conditions. They recognize the fact that their level of expertise may require supplementation by people who are experts in the field, so they don't have sufficient expertise in all of these fields. In fact, no regulator does anywhere in the world. But just to say that I have in the past taken these problems to the Department of Health when the MCC was still in existence, because people approached me with problems related to adverse effects. And unfortunately, nothing was done at that stage. I do think, however, that we have a much more receptive ear in SAPRA now than we did with the MCC. And so my recommendation would be that these should be reported to the MCC. And if it involves a practitioner who is clearly, in breach of all of the requirements from the HPCSA and elsewhere, it should be reported to the HPCSA.
Yeah, no, absolutely. I think we You probably need a guideline document about all the aspects, cellular therapy source material should be from A, B, C, D, E. Manufacturing should be in a facility that adheres to A, B, C, D, E. Patient administration should be in these facilities so that it's quite clear. Do you agree? Michael, do you agree? Sorry, maybe that wasn't clear.
Yes, definitely. Yeah, for sure.
Okay, this is, as I said, we have our cell and gene therapy meeting tomorrow. Oh, that's it. I was just going to close. Good for you. Please ask your question, Matt. Thank you.
Thank you for this wonderful talk. It's, sobering, not just wonderful, it's sobering. And it's good to see that you're having this discussion at a national level. I wondered, when I hear about all of the environments in South Africa as to what's going on in an unregulated way, I wonder about where the demand is. So which people and which individuals are seeking treatments that are not regulated, that fall out of conventional treatment? And I'm specifically wondering how much out of country demand there is. So as an example, are there those coming from highly regulated environments and very expensive environments, particularly Europe and North America, but perhaps now Asia and the Gulf, who see South Africa as an opportunity to obtain treatments that otherwise they wouldn't be able to get?
Thank you, Matt. That's really important. Can I answer that in two ways? So first of all, with regard to the need, so there's a demand because in many cases of chronic or untreatable conditions for which there is no cure, people become desperate, and they hear about this thing called stem cells, which is hyped beyond what is reasonable, and so they become vulnerable. And I get emails regularly. I've now actually started to put all of these together, and I can tell you that the main indications are things like blindness, diabetes, neuromuscular conditions, cerebral palsy. People are desperate, and so they hear about these stem cells, and all the emails that I get are roughly the same. We've heard about this. Do you recommend we go to this direction? Who can provide this in South Africa? Can we join a clinical trial? Et cetera, et cetera. So I think it's a question of desperation and a question of being profiled, these clinics being profiled as being a legitimate source of relief, which often is not the case. Then with regard to the notion of so-called stem cell tourism, I don't know. I can't answer because I don't have access to information in these private clinics. It may well be that people are coming from abroad and utilizing the opportunities provided by these clinics. However, let me just say that this notion of stem cell clinics in South Africa is not unique to South Africa. It is a huge problem all over the world. And there are major efforts being done in the US and Europe and certainly in Asia to combat this. So it's not a South African problem uniquely. So I suspect people would go to their local stem cell clinics first. But if they don't find relief and cost becomes a problem, South Africa could well be a destination. Although, as I say, I don't have access to first-hand information to be able to verify that.
Interesting that actually the majority of requests are not for hematological diseases, for which, though, we know that is the only really proven gene therapies. Even if you look at sickle cell, thalassemia, CAR T cells, hemophilia gene therapy. It's all in the hematology field, but the patients coming forward are in fact not in hematology for the most part. I'm not sure if that means we're doing a good job by saying what we do and don't have, or the fact that we actually do not have sufficient clinical trials in South Africa for these conditions. So on that note, if there are no other questions, I want to thank Michael once again for a fantastic session. And we know within the Cell and Gene Therapy Working Party, we have rigorous discussions about how to go about these things. And we have been working on a document for the last 6 months about how to say to the public and to our peers what is and isn't approved for these therapies in South Africa. So this has been very sobering, and we always love to know more in this area, and we hope to have more therapies available in the future and to regulate them better. So is there anything else from the floor? We had excellent attendance, Michael. We had about 150 people attend. Slowly people have gone off after the hour, but we still have 112 people on. So thank you very much, everyone, for staying. Thank you, Michael, for the excellent talk. And if there are any other questions, please feel free to contact Michael, since he is the expert. All right, and we hope to see you all at the upcoming conferences. Yeah.
Candice, thank you for leading this, the chairing and the questioning. And Michael, it was really good. And I tell you, the Cell and Gene Therapy Working Group, they rock. They are amazing. So let's continue.
Let's continue indeed. All right, have a wonderful afternoon, everybody. Thank you.
Bye-bye. Thank you.
Bye.